First of all, a nod to my co-authors and supervisors, Henry Rule and Daniel Jones. Eric Simon Ledo is not a supervisor. He was doing his PhD at the same time, roughly, as me. He beat me by a year, but Eric also very generously shared his data for his PhD, which allowed me to stand on the shoulders of giants and work with those data again. This concept of using data for more than one purpose was a recurring theme during the research and of course part of the transparency theme. Now, you'll see that in the title of this presentation, transparency is in brackets. That is not how it was originally sent to you. Just for questions of time, I've had to cut back on what I'm presenting. And so I'm going to focus on the monitoring side. And just for interest's sake, I'm going to throw in at the end of the presentation one slide on transparency. But I will make available this presentation in PDF format, and it has two sets of supplementary materials. So if I'm not an academic now, nothing is going to prove it to you. I have supplementary materials on the supplementary materials. So there we go. Without further ado, I'm moving on to the disclaimer. So as Diva mentioned, I do work, have a day job, but my day job has nothing to do with this presentation. So there it is. Again, acknowledgements to my two main supervisors, Henry Rule and Dan Jones, also to Eric for sharing those data. And initially, this was an idea I cooked up when I was still at the Institute for Advanced Sustainability Studies with Klaus Tupfer and Sebastian Unger. So many thanks to them as well for supporting me and getting this started. So I'm going to start with a picture of an old guy. This is Grotius or De Groot. I don't really know how you say his name properly because I'm not Dutch, but that was my go at it. And he was known during his time, by the way, on his treatise on War and Peace. So that's what you're looking at there. But strangely, history is a funny way of changing things. And he's now known best for Mare Liberum, which is his Freedom of the Seas treatise, which was done when he was quite a young man. And he states in it this idea that, and it was quite a reasonable proposition at the time, actually, that the sea is of such a vast extent that we can do with it as we please. And this was in response to legal attacks and physical attacks by the Spanish and English on the Dutch ships that were trying to trade through the Dutch East India Company. So he was hired as a basically a hired gun lawyer by the Dutch East Indies countries to fend off the English and Spanish. And he was saying, hey, listen, no one can own the ocean. It's all there. Basically it's a commons, what we would now call commons. And so this idea was one prevailing idea. 1609. 150 years later, this guy comes along, Jean-Jacques Rousseau, and he had a different idea about property and how things come into possession. And in the social contract, he talks about this idea, he talks about this idea that you don't truly possess something until such a time that other people agree that you possess it. So these are two very different perspectives. The first perspective was essentially finders keepers. Might is right. So if you can get that nodule, it's yours. In this view, it's no, it's only yours if society, if the rest of us, if the social contract agrees, it's yours. Two very different perspectives, and these two perspectives persist to modern day in the oceanic regime, where we have the water column that I would loosely say follows the Grotian regime, and we have the seabed, which, believe it or not, is following Jean-Jacques Rousseau. A book came along in 1965. This is a picture of my copy of this book. I own this book, The Mineral Resources of the Sea by John Moreau. You have probably all heard of it because you are all deep sea mining people. And it came about in 65. But I bet you haven't seen this quotation before. This is right near the end. And he writes, clearly the United Nations has no such authority. Now he's talking about deep sea nodules here. At the present time, therefore, the nodules would probably be considered the property of he who first reduces them to his possession. Now, of course, we don't use pronouns that way anymore. But more importantly, This is the Grotian view. This is the finders keepers view. We know that Ambassador Pardo had read this book because he quoted it extensively in 1967 at the United Nations. And so we know that he had read this passage, and he must have been furious. I can only imagine what he felt. But, you know, revenge was sweet because, as we know, it was no longer going to be a finders keepers regime. The law of the sea was negotiated from, I believe it was, let me see, 1973 to 1982 in what was known as UNCLOS 3. And so the finders keepers regime went to the what became known as a common heritage of mankind or humankind. All right, that's a bit of background to explain why we care. It isn't just finders keepers anymore. It is a social contract. It is an agreement by society that an entity has the right to exploit the nodules, and only under certain conditions. The protection of the marine environment is covered in UNCLOS, as we all know. I am not a legal scholar, and my PhD was not on the law, but I feel I have to include a few of these slides to set the context. UNCLOS talks about harm, and it talks about serious harm with regard to deep seabed mining. It also talks about this thing called the ecological balance. It's important to remember that biological diversity was not a common term, not at all used actually in the 1970s, and only came into usage in the early 1980s and did not make it into the language of UNCLOS. So UNCLOS has no biological diversity in it. It has this thing called the ecological balance, and that's about as far as you get. Now you notice that harm here is distinguished from serious harm. And it's an ongoing discussion in the ISA, which you should be concerned about, or both. But there's this thing called serious harm, which means that mining operations can be prevented or adjusted or halted due to serious harm. So the question is then, how do we monitor harm and serious harm, and how do we know we've crossed that border? And that's a recurrent theme in this talk. Now, this is what the ISA says serious harm is in its current draft of the exploitation regulations. At least, well, probably Hannah can tell me, they probably revised it since I last looked because they love to do this. It's probably even sillier now. But this is the, I didn't say that, I meant less, anyway, this is the current definition we have, which it says serious harm is a significant adverse change, and based on what we say and on internationally recognized standards and practices. Honestly, this doesn't help a great deal. We have to figure out what a significant adverse change is, and they probably didn't mean statistically significant, but maybe they did, because we don't know. What's interesting is when I went back to some of the foundation documents for the International Seabed Authority, and this is 1990. Now, remember, the ISA didn't come into being until UNCLOS was ratified and the Part 11 agreement came into power. So it didn't meet until 1995. But in 1990, the ISA Preparatory Commission had already been preparing, as the name suggests. And they had a definition for serious harm that you can see how the current definition arose from. But this one leaves much more for us to hang our hats on, our scientific hats, as it were. Significant adverse change, there it is again, there's terminology. But now they're talking about components of the marine environment. Now we're allowed to break it down into the physical and physical and biological components. And they talk about this thing called diversity, productivity, stability. So diversity has made it into the language now, you see? This language that wasn't in the original UNCLOS. So my observation here is simply that I think we had a better definition, at least in terms of giving us more to work with as scientists in 1990 than we're currently negotiating today. But I leave that open. That's not the central point of my thesis. This is simply some background information. But so whatever the serious harm thing is, we're going to have to measure it in some way or other. And this is monitoring environmental harm. Now, one thing I do put forward in my thesis is that we cannot wait for harm to get serious. We have to measure it before it gets serious. Now, when you think about it, that's pretty self-explanatory, but the regulations are not necessarily walking down this road right now. So I think we have to make a very strong point to scientists that by the time things get serious, it's too late. And we want to be able to spot harm to the marine environment. And indeed, it's an internationally recognized good practice to be able to monitor harm before it gets serious. And I call it almost serious harm. So how do we monitor almost serious harm? Now, this question of monitoring the deep sea has become a little bit controversial. Look at this new paper that came out, and look at the size of that font. It has nothing to do with me filling the screen with it. Ecological variables must include microbiota and meiofauna. Oh dear, oh dear. And look at all the authors on this paper. Some of you are probably on the call right now. What is this all about? Well, this was about a paper that came out in February this year. So this one just came out a few days ago. This one came out in February by Danovaro et al. And again, some familiar names on that list. And honestly, the paper does recognize Mayo and microfauna, but the abstract really doesn't do a very good job of recognizing them. This quotation is taken from the abstract, and it talks about large organisms, macro and megafauna, and it talks about trophic structure and biomass production. Now, this is craziness because I'm pretty sure none of the authors, or very few of the authors, had read my paper that came out before this, but this is actually what I was talking about. I know the microfauna people are going to hate me for the moment, but I looked at megafauna, and I'm going to talk to you about trophic structure and biomass in a moment. Biomass production is a little different than biomass or density, but we're kind of in the same ballpark here. And trophic structure, I'll get to in a minute. And I did look at megafauna because the data were available, and that was Eric's data set that I mentioned earlier. It's rather easier to survey megafauna, through remote vehicles and so forth than it is some of the other stuff. And so the data were available, it was readily surveyable, and that is what I'm looking at in this presentation. That said, I do recognize that the world is more than just megafauna, and megafauna in this case were greater than 1 centimeter in length. Strangely enough, the ISA guidance sets 2 centimeters for megafauna, which is a very odd cutoff. We looked into the literature, and I think Fred Grassel on up always talk about 10 millimeters. So I'm not really sure where the 20 millimeters came from, but we use 10 millimeters as our cutoff, according to what's generally accepted in the literature. And here's the paper that is one of the core pieces of the PhD research I worked on, the chapter that took more than 2 years to write. Yes, thanks for nothing, Daniel, for sending back all those edits, but few have read. I think it's a good paper, honestly, and that's why I'm going to present it to you here, because it's sort of I think it's viewed as too niche or something. I don't think many people have read it. I'm not getting huge numbers, but I think it's incredibly important, and I hope by the end of this presentation, you'll understand why. So I call it the statistical imperative. When monitoring, whether we're monitoring microfauna, meiofauna, or macrofauna, or whatever, we need to be talking about statistical power analysis. And you're like, what? What? Seriously, Jeff? And I'm like, yes, seriously. We need to talk about this. This is a dirty little secret. A no effect result without enough statistical power tells us nothing. And worse, it is misleading. It provides a false sense of assurance. I know that you guys all know this, but no one's talking about this, and I'm concerned. I'm very concerned. Here's why. 3 environmental impact assessments have been submitted so far to the International Seabed Authority. I'm going to go through them one at a time. The first 2, the ones from BGR and GSR, I believe have been accepted, though it's not 100% clear, but I believe these 2 have been accepted. So this is the first ones from BGR or BGR from Germany, and they were not getting results. They were finding things like they only had a third of the species that were common to the PRZ and IRZ, and they're postulating why. And they say that, well, maybe the amount of analyzed samples is too small to obtain an accurate picture of the entire community. And then they go on to say it's still difficult to estimate the number of samples that would be required to reflect the whole community with any sort of statistical significance. So I must hand it to the Germans, and as I think most of you know, I lived and worked in Germany for a while. They were being straightforward here and they were being honest. They're just basically saying they don't think they had sampled enough and they're not sure how much they would need to sample. Well, thankfully, I've done this research, so now I can tell you how much you need to sample. But there it is. So this is one of the EIAs that was submitted and I believe accepted by the ISA, and they're basically admitting in it that they've undersampled. Here's the next one. This one's from the Belgian company, Belgian-based company, CSR. And they're talking, it's a little harder to understand what they're talking about here, but they're basically saying that Shannon Weiner and Pilot are significantly affected by this thing called expedition and so on and so forth. What are they talking about? What they're talking about is their two sampling trips, the expedition variable, was by far the single largest defining factor. There were no other impacts detected other than the fact that when they sampled on two different occasions, they got two very different results. So this suggests, we don't have the details of the sampling regime in this EIA, but it very strongly suggests that the sample sizes were too small. And it was the same problem that we are having, that the Germans were having BGR previously, which is too few replicates to attain a sufficient, overview of the situation in the deep sea. In other words, insufficient statistical power. It's looking very strongly like that. Let's go to the third example. India. Here, the Indians take a different approach. They found, weirdly enough, in some of the restoration sites, that biodiversity was bigger than before the treatment, and that basically nothing was adding up. And I should also add their Pilot's evenness is a very telling, almost exactly 1.0, which tells us that they're almost entirely sampling singletons. And I go into why. I do a bit of reverse engineering in the thesis to show how this might be. But they say, no, this is probably due to underwater natural disturbances, and go on to say that it was natural variability that affected our results, and that the initial effect of the disturbance experiment has waned off, is their wording. In other words, the disturbance is gone, and we're just looking at natural disturbances. Now, of the three examples I've just provided, I think we've gone from the most straightforward to possibly the least straightforward. There is nothing in the Indian EIA that suggests this at all, that warrants this conclusion. There is nothing to show that the effect of the disturbance has indeed waned off. Rather, they're postulating this because they weren't getting statistically significant, or I want to use the word meaningful results. Significance has nothing to do with it. It's meaningfulness, and they weren't getting statistically meaningful results, and therefore postulate that it has to do with they must have gone away. I would suggest, as you can tell, there's this pattern here. I would suggest they lack statistical power. And if you look at actually their sampling regime, they only use 300 photographic frames in their analysis. This is ridiculously low. And the analysis that I'm about to show you, we use 10,000, or Eric's hard work produced 10,000 different images that were analyzed. So 300 versus 10,000, you guys make up your own mind. So it appears that all three EIAs submitted to the ISA to date have lacked the statistical power to detect the sorts of impacts, and that means harm, that they were supposed to be monitoring. So whether the answer is myofauna, microfauna, or megafauna, it doesn't really matter if you're not monitoring them properly. And I'm very concerned about this. And I feel that this is not being adequately discussed in the ISA. ISA uses wording in the LTC comments like statistically robust, but then does not go on to define what that means. And I think we need more explicit guidance on what statistically robust means, because we're not seeing it as of present. So, this should be a review for you, but there's basically three questions that we look at when we try to figure out adequate statistical power. How large of a sample we should be taking, how many replicates, and then what are we trying to detect? question 3. And remember, I'm talking about this idea of almost serious harm, harm before it gets serious. How are we going to detect things are getting worse before they're too bad? And this third question is actually the first question that has to be asked, because we cannot set up a proper experimental monitoring regime until we know the level of the effect that we're trying to detect. Now, I know that sounds basic, but believe it or not, that's what we're doing right now. We don't know what we're trying to detect, and we're already down the road of creating rules and regulations, and we're already actually approving EIAs without knowing what it is we're trying to detect. And this question of how much we're trying to detect is not just a question of science, though it definitely is. It's also policy, law, and politics. And any of you who've been involved with the ISA know that politics play a role. And it needs to be answered. We need to have an answer to this, at least a range of values, before we can proceed on setting up sample size and numbers of samples. So we don't have an answer to that. And so the research I'm presenting basically postulates what it is we might want to be detecting, and then looks at, if we were trying to detect that, what we'd need to do. So everything that I'm about to present will still need to be given it will still have to be agreed to in the international forum known as the ISA. It will still need to be sorted out one way or the other. So this is what we did. We took megafaunal photo data from the CCZ, essentially because, as I said, those data were available. It is very, very hard to get deep sea mining data, as I think we all know. But thanks to Eric and Dan, who is Eric's supervisor. And we virtually killed organisms in a series of simulations. We basically took real data, and then I started weeding them out. And I weeded them out based on 2 different simulations of stresses. One that impacted organisms based on nodules, the idea being that many of the nodules will be sucked up, and therefore, and then, well, actually sorry, let me try that again. This simulation is based on areas next to mining. The assumption here was that mining itself would be extremely impactful in its immediate footprint, and none of these sort of fine-tuned questions matter much. But what about the areas next to mining operations? When do we start to see almost serious harm? So one hypothesis, as it were, was that those suspension feeders largely associated with nodules would suffer more than the burrowers and so forth. And so they might suffer more. So we were killing off those animals based on their affinity to nodules. The ones that only lived in nodules got a higher hit rate than those that were only sometimes found around nodules. And then the second stressor that we applied was spatially random stress to everyone. And this is the idea that plumes, of course, are chaotic, and they will impact some places chaotically more so than others. That's just the way it works. And and so then we added those stresses together, and we looked at what the overall impacts might be. And it was a pretty simple numerical model, honestly, but it gave us results. We also looked at 8 different ways of measuring the impacts of biodiversity. I'll get to those in a minute. For those of you who are not that familiar with things like the CCZ, this is a typical rank abundance distribution that you will find. Pretty log linear in the middle bit. There's an interesting, though, inflection at the top decile. So there's the top decile. There's a lot of very abundant organisms, or very abundant, relatively abundant. Sorry, nothing's very abundant down there. Relatively abundant. And then we have at the other end the extreme number of singletons, doubletons, and tripletons. So, it's amazing, but in Eric's research, with 10,000 images, it's incredible to think that more than 1/3 of them were just singletons, doubletons, and tripletons in terms of Morpho species. And he was looking at Morpho species. This is really an astounding finding, that down in the deep sea, It's not like on land. It's not like in the shallow ocean. It's very sparsely populated. Diversity is high. We all knew that, but abundances are low. And some of them you only see once, twice, or three times, even though you do extensive sampling. So this makes it statistically very tricky to tell when you've reduced one of those species because they're already in very low numbers. And I spotted this right away, and this is why I wanted to do this research, because I thought, wait a minute, no one's talking about this. This is a different world down there. Statistically, it's a completely different world than we're comfortable with up here on land, breathing air. These are some of Eric's unpublished species accumulation curves when they were just exploring the data. The important thing to note here is there is no flattening off. And I think all of you who have researched in the deep sea have experienced the same thing. There is no asymptote. We're still counting species. 5,000, I think on the x-axis, if I remember correct, is how many kilometers of transect he looked at. So after 5 kilometers, not 5,000, but after 5,000 meters, 5 kilometers, it hasn't flattened off. It's still going up. We're still finding new species, or new morpho species, I should say. And he broke it up into flats, ridges, and troughs, which I won't be presenting here, but I also looked at that in my research. So yeah, the asymptote not reached. That's what I just said. Yeah. Here are the 8 metrics we looked at. Density, which is just a corrected measure of abundance. The geometric mean, which is used in some situations. Richness, I'll get back to richness. Exponent Shannon, inverse Simpson, those are Hill number corrected versions of those familiar measures. Pilot's evenness, this was the dark horse, I'll give it away. Didn't think Pilot would be that interesting. Turns out it was. Fisher's alpha, I was convinced that Fisher's alpha was going to be the dark horse. It was based on a logarithmic assumption of species distribution. And I thought, that is exactly what we have in the deep sea. Fisher's alpha is going to save the day. It didn't. And the Chow 1 estimator, which again, it relies heavily on singletons. It looks at the singletons and data sets, and I think doubletons as well. And I thought, oh, this is cool because it's just like that down there. But actually, the Chow 1 estimator didn't do as well as I'd hoped either. So, moral of that story is, don't know till you try. This is an example in bar graphs. I'm going to later, next slide, show you even more complicated diagram. This is trying to ease you into it. This is showing the effect of different treatments. In this case, on 750 individuals, starting point. And then we started beating them up, virtually murdering them. And what happens? And we have the X and Z axis are the 2 different treatments that were added together, and then we decided if the animal lived or died. This point right there is what I'm going to be here on in calling the tipping point. You can see on the back row that there's not much effect of those treatments. The numbers for richness and abundance are pretty constant. The top number is abundance and the bottom number is richness. Yes. But you can see that things start to go pear-shaped right in that middle column. So this is what I'm calling the almost serious harm. Things are starting to look bad right around there, and all the other columns get significantly worse. Now, I will tell you that this didn't come easily. We trialed lots and lots of scenarios until we got a set of scenarios that could demonstrate this point. So this is a simulated tipping point. Now, this is going to be the most difficult graph I have to present in this presentation, and so please bear with me as I try to explain it to you. First of all, this is a panel diagram. So each panel is just showing a different situation under the top, the rows and columns. The rainbow colors represent different treatments. And if you look under in any particular panel and set of treatments, you'll notice that they decline, they're going down. So this black square at the top of each panel, you see those black squares, that's the control situation, the situation before we started killing organisms. And then we start applying different treatments, and you can see their numbers go down, or whatever it is. we looked, we looked at samples of 250 individuals, 500, 750, and 1,000. I'm only showing up to 750 because the differences between 750 and 1,000 weren't very much. There is an asymptote there. There is a diminishing returns. So looking at these, and in this particular panel, remember, oh, sorry. Remember, we had 8 variables we're looking at, but if you look at the rows here, it's just density and richness that is being displayed here. What I've circled on the right-hand side is 750 individual sample size, looking at everything mixed together. We also looked at nodules, habitats that had nodules and habitats that did not have nodules separately. So you'll see those are the separate columns in that panel. And not shown here, we looked at troughs, ridges, and flats to see if those geomorphic characteristics of the habitat affected things as well. There's been this idea that we may need to do stratified sampling to get a proper view of the environment. And so we looked at that geomorphology to see if that was true. Short answer is generally not, but sometimes yes. Just for simplicity, we're going to look at everything mixed together. So we haven't stratified the results at all. The nodule environments have been mixed with the no nodule environments. But before I go to that slide, just notice how different the no nodules results are from the nodules in the all. The no nodule is a more depauperate, if I may say, environment. There are fewer, there's a lower density and lower richness right off the bat. Now, you biologists probably knew that, but statistically that's important because it's very, very different from the nodules environment and the mixed results, which are dominated by the nodules environment in general. Okay, so I'm going to try to explain a little bit more what we're looking at here. But basically what you have to take away from this is one of a huge series of panels that we did looking at how we could beat up the species under different regimes of treatment regimes. It's a nice, polite, scientific way of saying killing them, and seeing what the results would be and if we could measure those results. The whiskers here are, the error bars are this 95% range. 95% range is not statistically the same thing as 95% confidence level, but think of it as being almost the same thing. So we're looking at the 95% confidence range of the results in the error bars. So clearly here, do you see I've just put an arrow on? That is our tipping point. That is that other column I showed you before. And in terms of density, it's clearly outside of the error bars, the other treatments. So you'd be able to pretty quickly identify this treatment under density. So it's important to see, whereas the earlier treatments, you can see everything's within those error bars. It'll be harder to discern them. And likewise later, when things start going pear-shaped. So I've taken a little bit of extra time to explain this graph because this graph is essential, these charts are central to everything that we did. Yeah, and you can see I put another arrow here under richness. Under richness, it's actually really hard to detect the tipping point. Look at those error bars. They're overlapping the other treatments quite a lot. So turns out, surprise, surprise, richness is a pretty crummy indicator, and I'll explain why. So that first graph I just showed you, maybe I'll just go back. You can also see that the error bars get more compact, smaller, as we increase the sample sizes. So from 250 to 500 to 750, we are reducing our, so to speak, error or our range of results. So actually, I'm getting ahead of myself, but we recommend around 750 individuals is a good sample size to start with. 500 to 750. Eric recommended 500 in his research, but remember that's because he wasn't killing anything. Once you start removing animals, then obviously you have fewer to measure. And so I'm suggesting, wait a minute, we got to start with more than 500 because they're going to be affected by the neighboring mining operation. So taking that into account. This slide is about how many replicate sites do we need, and it's power analysis. The x-axis is logarithmic. It goes up to 32 replicates, and then I cut it off there because I can't imagine contractors wanting to do more than 32 replicates. In fact, I think they probably want to do a lot fewer. But so we only looked up to 32, and then I just chopped it off there. I will explain this in more detail in a moment, but again, the lower row, what's called 25N, 26A, which really does mean something, is that tipping point treatment. So the earlier one, which is just a lesser impact, you notice that most of the bars, and this is all you need to really notice here, most of the bars are exceeding 32. So in other words, to detect a minor or a smaller impact will require, in many cases, more than 32 replicate sites, which is amazing. This is really bad news if you're just trying to detect a fine change, because you're not going to without huge sampling effort. The only thing that picks up these changes early in the game is density. Notice that's the red columns, and under 750 individual scenarios, 8 replicates or so will probably do the job. So if you're trying to detect smaller changes, 8 replicates under density. Okay, I'm going to look a little closer now at some of the other measures. Oh yeah, I wanted to point out density does really well at the tipping point. You only need about 3 replicates in this particular scenario to detect it.
Jeff, just a quick reminder, we have about 19 minutes left in the session. So if you want
19, okay. Thanks for the reminder, Diva. I did ask you to give me that reminder. So what I wanted to point out is Pilot's evenness needs 5 replicates. Inverse Simpson, which is very close to exponent Shannon, needs about 10. Richness needs 24. And why is that? Because richness, of course, one animal counts. Richness is a really bad measure. If you're trying to get an early warning, richness shouldn't be used, but people do use richness all the time, but it shouldn't be. It's not going to give you an early warning. Richness relies on animals being completely removed from the sample before you can detect it. So this just shows that. But the surprise here is Pilot. Pilot, which is a measure of evenness, generally considered a measure of evenness, can actually be considered a measure of trophic structure if you're looking at changes in Pilot, changes in evenness. And Pilot comes out really well, and I could discuss why, but don't have time. But 5 replicates to detect the tipping point simulation under Pilot. So 5 replicates to see that the trophic structure, or the structure, community structure, excuse me, not trophic, community structure of the megafauna has changed. Weird little thing here. If you measured the non-nodules and nodules together under richness, you'd need about 15, 16 replicates. But if you mix them all together, you need 24. There's a reason for that. I don't have time, but there it is. This is the Cohen's d, the measure of impact. The more sensitive the measure, the bigger the impact. That makes total sense, but people haven't acknowledged this. So the statistical impact varies depending on how you're measuring it. And richness, as you can see, has very low impact because it is very low sensitivity. So what do we need to detect? What do we need to detect? Well, Dan Jones et al. Noted that historical experiments had a Cohen's d of about 1. And in my simulations, it goes from 1 on up to 3. I'll just go back to the previous slide. 5, 6, notice some of those numbers. 5 and 6, 8, that is unheard of in other literatures. A Cohen's d of that size is just like massive, like unbelievably huge, and would definitely be considered serious harm in another regime. And here it's just something we're just starting to detect. So we can't really determine what monitoring regime we need until we know what we're trying to detect. And I've made that point before. But it's probably going to be much bigger than what we're used to being comfortable with in other environments, because these numbers are much larger than normal for effect size. This is a summary of the results. Sample size should be 500 to 750 individuals, at least 5 replicates at least. The early warning metrics came out, density and Pilot. So there it is. And please, please, please do not test for power at 80%. I don't know why that's considered a standard in the literature. Please test your power to 95%, just like we do significance. So Before we get meaningful regulatory thresholds, we need to bear in mind that the sample size, the replication, and the metric altogether do affect the measured effect size. So we can't set a threshold size until we have set the experimental regime. This is bad news, actually, for regulators, but it's something we need to know. So in the upcoming ISA standards and guidelines, we will need to consider these things, indeed the measure being used, before we can set a proper threshold. These are three sort of take-home messages. Please ensure environmental data are readily available. We all have opinions about the current database available from the ISA, but I would say that DSM data are not yet readily available. Establish robust statistical practices. That's what I've just spoken about. And please be inclusive when considering the results of data analyses. We need to be open and inclusive as well as rigorous when discussing about what constitutes almost serious harm. And here's a little PS. The transparency of the ISA compared to regional fisheries management organizations. This was in a 2016 paper. ISA's in red. I'll let you decide if you think the RFMOs are very transparent, but the ISA did not fare very well next to them.